Good afternoon. I am Julio. I'm very honored to serve as the UCLA Chancellor, and I'm especially honored today because today is really a very special day for UCLA because we are privileged to welcome Fred Ramsdell back to his alma mater, back to his home. His work, as we all know, has shaped modern immunology. His career reflects the very best of what comprehensive research universities like UCLA are capable of. When the Nobel Committee reached him with the news of his award, as I'm sure you have all read, Fred was hiking with his wife in the Rocky Mountains, very far from phones and from headlines. And there is something fitting in that image. Here you have a scientist absorbed in the world, following curiosity, even as the world prepares to celebrate his work. Dr. Ramsdell's return to UCLA as a Nobel laureate gives us an opportunity to recognize his achievements and to learn from him. In today's seminar, he will share insights from a body of work that has influenced how we understand the immune system and how we approach disease. This recognition comes during a remarkable year for the University of California system. UC faculty and alumni received 5 Nobel Prizes across medicine, physics, and chemistry. It is the most ever awarded to a single institution in 1 year. And that achievement just reflects the sustained commitment across all 10 campuses that form the University of California system, our commitment to education, to research, and to collaboration. UCLA has long, we're very proud to be part of that tradition, and gatherings like today's remind us of how central this is to our daily work. Because research powers progress. This is the tagline we've been using at this time of attacks on science. Research powers progress. It expands understandings and opens new paths forward. In immunology, those paths affect real lives. Dr. Ramsdell's discoveries have helped reshape thinking about autoimmune diseases such as arthritis, multiple sclerosis, Crohn's disease. And they have influenced approaches that continue to guide research, clinical trials, and treatment strategies around the world. This work also speaks to the future we are building at UCLA. As part of our strategy, we have developed something we call the UCLA Research and Innovation Blueprint, in which we're developing and strengthening our innovation ecosystem across Southern California. And this blueprint is inspired on a strategic conception about what it is to be a comprehensive research university. And it's the simple proposition that universities have one product, only one product, but it's a very powerful product. It is knowledge. Knowledge is the most powerful force for enlightened social transformation. And we engage in that product through an integrative circle of knowledge. We start with the creation of new knowledge. That's what basic research does. It continues with the recreation of knowledge, which is what good education does. It's not just transmission of knowledge. It's recreating in the minds of the next generation. And then, critically, it proceeds with the translation of knowledge. Translation in 2 forms: into technologies, drugs, vaccines, iPhones, everything that we have that has improved the quantity and quality of our life, which starts with basic research, translation into technologies, and then translation into evidence that guides decision-making. And when translation is effective, knowledge can be applied or utilized. When we do that, we change the world for the better, and the circle starts anew. And that's the vision as a comprehensive research university. One key resource we now have at our disposal is the UCLA Research Park, just a couple of miles from here. It is anchored in areas where UCLA leads, including immunology and immunotherapy. And it will bring together researchers, clinicians, and partners from industry to move ideas from discovery to impact, from science to solutions. Dr. Ramsdell's work reflects exactly this kind of comprehensive inquiry that then gets translated into benefits for society. To finish, I want to add, because I know many of you here are faculty members, that scientific progress is really shaped by learning communities and by mentorship and the importance of mentorship. During his doctoral studies at UCLA, Fred was mentored by Sidney Golub, now a professor at UC Irvine. Professor Golub's guidance reflects the quiet but powerful role that faculty play in shaping how young scientists think, ask questions, and pursue their work. When a brilliant mind wins a Nobel Prize, those who nurtured the laureate share in the pride of that accomplishment. In a few moments, our department chair, Dr. Hector Aguilar-Carreno, will formally introduce Dr. Ramsdell. Before that, I just want to thank all of you who contribute to UCLA's research mission, to our faculty, our staff, our students who create the environment where discovery happens and where talent is nourished so that it can flourish in the types of achievement that Fred personifies. You sustain the culture that allows ideas to grow, sometimes over decades. I am deeply grateful to you and to everyone who helped organize this conference. And one of the hardships of my job is that I get to miss the very most interesting things like today's lecture, but I will watch it on video. So I will be departing soon. I did want to be here to honor Fred and to welcome you back home. This is your home. We're immensely proud of you. Thank you very much and enjoy the rest of this wonderful session. Thank you.
Good afternoon, everyone. I'm Hector Aguilar-Carreno, the chair of the proud Department of Microbiology, Immunology, and Molecular Genetics, MIMG, formerly MNI, Microbiology and Immunology. It is a rare honor to introduce a speaker whose work has quite literally rewritten the textbooks of modern immunology. Dr. Fred Ramsdell is a visionary biotechnology leader with a career spanning over 30 years, a journey that has taken him from receiving his bachelor's degree from UC San Diego to the labs at UCLA as a Ph.D. student in our department, to the NIH as a postdoctoral fellow, all of which led him to the helm of industry-defining organizations like the Parker Institute and Sonoma Biotherapeutics. In our field, we often talk about pioneering research, but Fred has defined it. While at Darwin Molecular, his team discovered FOXP3, the master switch of regulatory T cells. His discovery was so foundational to our understanding of the immune system that it was recognized with the 2017 Crawford Prize, and most recently, of course, the 2025 Nobel Prize in Physiology or Medicine. Fred has spent his career translating complex science into life-changing therapies, navigating the rigorous path from discovery to the clinic at institutions like ImmuneX, Cymogenetics, and Novo Nordisk. Today, he joins us to discuss a fascinating evolution of scientific endeavor. I want to thank the California NanoSystems Institute, CNSI, the Division of Life Sciences, and the David Geffen School of Medicine for partnering with us, with our department, in sponsoring Dr. Ramsdell's visit, and my assistant Pablo Morales for organizing this whole visit and event. Please join me in welcoming Bruin alum and Nobel laureate Dr. Fred Ramsdell for his address from the Manhattan Project to Autoimmune Therapy.
Thank you very much. Okay, I don't know if we showed this or not. Did we show this? Okay, I'm sure you've all seen it. This is new to me. We didn't used to do this. Okay, so now I have to figure out how to make this work. Okay, here we go. Okay, I said I was going to walk around, but I don't have a clicker, so I'm going to walk back here a lot. First, I want to thank the Chancellor who left, and if I If I meet someone who's brilliant, I'll make sure to pass that along to him. I want to thank, obviously, Hector and most importantly, Pablo, who did all the work around this. Nothing personal, Hector, but I'm sure Pablo did the actual work. And everyone else for coming, the deans, everyone who invited me back. It's, I've been back to UCLA a number of times over the years, but I've always been whisked into a medical center building, a conference room, spent the entire day there, left and went to dinner. So I haven't actually been on the campus since 1988. So it's changed a little, which you may not know, but I certainly know. People said, have you been here before? I'm like, I don't know where I am. So I don't know how to answer that question. But it is great to be back. I had a great 4.5 years here getting my Ph.D. Yes, it was 4.5, so if any of you are in your 6th year, get going. But it was really impressive, and I will say, I don't think I appreciated what the education was like when I was here. In fact, I'm quite sure I didn't. But when I got to the NIH, which was probably the center of the world for immunology research in the late 80s, I realized that I actually knew what I was doing. I was actually well trained. I had a lot of opportunity here to work with various people in various areas. And I could see some of the other postdocs coming into the NIH and realized I was trained better than they were. This is great. So it was really, a lot of my ability to succeed, I think, started here. Really started with understanding the fundamentals of immunology and how they worked, and how to work with people and collaborate. And that's a theme for me that's been really, really important. I went into biotech, as you heard, instead of academia, because I actually very much like the collaborative nature of biotech, and I'm happy to talk with people about that later. But when I started in my postdoc, certainly and in science, my presumption was that people who went into biotech were the people who couldn't survive or succeed in academia. And I can tell you that that's, aside from the medal thing, that's absolutely not true. The talent level is spectacular as long as you're aligned on the goals and what you want to get out of your career. So I've had a fantastic career, and I really did all start here. So, go to this. Okay, so disclosures, you'll see a bunch of Sonoma. That's the most relevant one in a second. So how do we get into this? So in the early 90s, early to mid 90s, I and a number of us became very fascinated with what mouse mutations had taught us. And these are a bunch of spontaneous mutations. But what they taught us about the immune system, there's many others as well. And we were just really at the beginning era of doing knockouts in transgenic mice as well, single genes, figure out what those do. And we sort of naively thought if you knock out a gene, it'll tell you exactly what it does. And that's somewhat true, but not completely true. It's obviously more complex than that, and I think we all appreciate that today. But we've learned a lot from these spontaneous mutations, and what these things do was they led us to genes and pathways we didn't know anything about. And for someone who really likes discovery, this seemed like a really interesting path. And so we built programs to not just harness the spontaneous mutations you could find, but to generate our own. So we built ENU mutagenesis, or a large ENU mutagenesis program to create our own mutants. I won't talk about that today, although there were some interesting things that came out of that. And other groups would do the same thing. But based on this kind of background, we had the fortune to be supported by Bill Gates and Paul Allen, people you've probably heard of. And they were in the business of supporting what was at the time Darwin Molecular, not to make money, because they had more money than they knew what to do with. What they wanted to make was a change. They wanted to make a difference. And so they helped support us, and one of the programs they would support was the genetics program, which is not a very clear path to a drug or to a way to make money. And yet, they were willing to support that. Not something that's common today, but I will still put a plug for the concept, because I think you can find really important nodes of biology no matter how you want to study them. But we had these, but we didn't have, this list isn't endless. It doesn't go off the board, and there's not a huge list of mouse mutations to study that we felt were really exciting. So we went looking. And fortunately for us, the CEO of the company used to run the Department of Energy's genome program. And he knew about this group at Oak Ridge National Labs. And so Oak Ridge National Labs was a this is a facility, but it's really a town that was built in Tennessee during the 40s in secret. So people nearby didn't even know this town was existing, except that they would see all these vehicles go out and they would see construction and stuff, but they didn't know what it was. And the reason they didn't know that was because it was there to figure out how to make the bomb. So this was part of the Manhattan Project. And as you can see, the real goal here was to enrich nuclear material to be used in atomic weapons. And in fact, it was used for atomic weapons in Japan. Now, one of the other things that the government decided was if we're going to make radioactive bombs, we should probably understand what radiation does to mammals. So they set up a mammalian genetics unit at the same time, which I find shockingly prescient for our federal government. Not just in current times, at any time, to be perfectly honest. And they hired Bill and Leanne Russell, who came from Jack's Labs and were preeminent mouse geneticists at the time. And you can see the mouse racks there in the background, et cetera. And their role was to really understand how ionizing radiation and other mutagens affected the genomes of mammals. And they did this in a variety of ways. They would expose animals to varying levels of radiation and figure out what the damage was, et cetera. So all the radiation tests that limit our exposure came from these experiments in the 1940s. That was the genesis of all of this. They invented things like the specific locus test and many other things that would allow them to measure the effects of ionizing radiation across the genome. They literally would put mice in their Volvo. They would drive to White Sands, New Mexico, place them at varying distances from a bomb. They go into a little shelter, bomb goes off, collect all the mice, drive them back to Tennessee and see what happened. Not the most quantitative way of doing it, but they certainly got data. And we found out about, in conversations with them, about a particular mouse, a relatively famous, now famous mouse called Scurfy. And this is what it looks like. It has truncated ears. Its eyes are basically closed. Its fur is all mottled. It has essentially every autoimmune disease known to man. It has psoriasis. It has IBD. It has type 1 diabetes. It is a very, very sick animal. The gene responsible for this is on the X chromosome, so it's half the males within a litter will come down with this disease, and they will die at about 3 weeks of age. And so that seemed really important to us, and we thought, okay, this is a node of biology that we should understand because this is really key. Now remember, they made this mouse in 1949. Oh, and by the way, this was a spontaneous mutant. This never left the facility, was never exposed to radiation. So it's good to be lucky, I guess. Not for the mouse, but for us. It's good to be lucky. But they kept this strain alive since the 40s because they knew this was really important. But they did not have the ability to figure out what the gene was, right? They just didn't have the tools. So they did a lot of characterization. You can see people here, Virginia Godfrey and Irby Wilkinson and others, who would characterize these animals and demonstrated that it was a T-cell mediated disease. It was predominantly a CD4 T-cell mediated disease. They characterized a lot of the inflammation, et cetera. But beyond that, they didn't really know what the problem was. So we got these mice and said, all right, it's the mid-90s. We can clone stuff. We can sequence DNA. We can do this. The human genome had not yet been sequenced, and so sequencing was still in its infancy, but we could do this. And this is just a representation of the T cells in these animals. And I won't go into that. I'm not going to go into any great scientific depth here, so if you want to do that, we can talk later. But I'm not going to do it in the talk here. But essentially, the T cells from these animals were hyper-responsive. They responded much more easily and to a much greater extent than did T cells from a normal littermate control, a wild-type animal. And they only required one signal, which was unusual. Most T cells require 2 signals to actually go. These guys did not. They just needed one. And didn't matter how you measured it, they would respond incredibly fast. So we saw this as an opportunity to understand tolerance, because this animal clearly was not tolerant to its own tissues. And so if you dial back to the beginning of the century, the early 1900s, it was postulated that while humans can, and large animals in general, can respond to the pathogens around us, we don't respond to our own tissues. So we make this incredibly diverse repertoire, this incredibly diverse ability to respond to almost anything, even inorganic molecules we can respond to, but we don't respond to our own kidneys and our own livers and everything else. And so there's a control mechanism, and that was postulated as tolerance. And people began to develop data to support that in the 1940s, dizygotic cattle from Ray Owen. McFarland Burnett and Peter Medawar won the Nobel Prize in 1960 for describing this in much more detail and providing evidence to suggest that there are mechanisms that actually eliminate cells that see our livers and our kidneys and other things, but still allow the diversity that can see all sorts of bacteria, viruses, fungi, whatever else. And so they won this in 1960. By the way, 60 was the year of my birth. They gave their lectures 2 days after I was born. I just found that kind of interesting. So we thought this is a really good way to understand what's going on in tolerance, because we know it must be broken in these mice. So the problem was the genetic region was too big for us to sequence back in the day. It was 3,300 kilobases. And while today that's not much, in 1995 that was a lot. So we couldn't do it. And we were actually pretty good at sequencing. We'd identified a bunch of genes and a bunch of other conditions, but they were from smaller genetic regions. So we need to narrow it down. So you narrowed it down by doing a breeding, and we didn't have an animal room, so I confiscated a janitor's closet. And I still don't know whether we actually told the janitor. But we took the janitor's closet because it had a sink. And the reason the sink was really important was we had to wash a lot of poop out of a lot of cages. So we could wash poop out of cages and put in the back in. So we set up a back cross in this janitorial closet. I'm also 99% sure we're not allowed to have mice in this building, but I didn't ask, we just did it. And so we did condense this region, and actually, when I say most of the all of the genetic work was done by a woman named Mary Brunkow. Mary is the person I shared the prize with. She was the geneticist. I was the biologist on this program. And we worked hand in hand throughout this whole process. And so we narrowed it down to 20 genes, or 20 open reading frames. You'll notice some of these have, ridiculous numbers, AA273, blah, 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 right? So it looked like an open reading frame, but no one knew what it did. It didn't have any motifs. It didn't have any sequence similarity or homology to anything we knew anything about. So there were 20 of them. And so I prioritized these 20 and said, okay, we have to sequence the mutant version and a version of the gene that's wild type and hope that there's a mutation in a coding sequence. Because if it's not in the coding sequence, we'll never find it. And we're just going to walk through them one at a time until we find the right one, which went on for months and months and months. Now it would take an hour, then it took us 3 months. But we found it in the last gene, in the 20th gene. So as I tell people, I could not possibly have been more wrong than this in prioritizing this. And it was a gene that was called, that we called FKH, which stood for forkhead, which meant it was a forkhead containing domain in it, which meant it was going to be a DNA binding protein at the end of the day. And our ultimate goal, this is all interesting, but our ultimate goal is to try to understand this node of biology so that we can understand human disease and maybe, treat somebody. That was the idea. Transcription factors are a lousy way to try to get at that process. They're not a great way today, although there are options. They're a terrible way in the mid to late 90s. So that's one of the reasons it was at the bottom of the list. And there's a couple others I'll come to in a second. But that was the gene, and this is what it looked like. I love showing little old notebook pages. This is the way we used to do stuff in the old school. So this is April 9th, 1998, and you can see that there's a two base pair insertion in this gene, which leads to a truncated protein, a truncated non-functional protein. So, all right, so this isn't formal proof we have the right gene, but we're still pretty confident. It's the only mutation we saw anywhere in this whole process. So, yeah, we have to go do the formal proof, which we've done, and you'll see some of that. But we're pretty confident that this is the right gene. So that was super exciting. Eric Jeffrey walked into my lab, or my office rather, and said, we got it. And he shows me this, and I'm like, that's awesome. Crap. What do I do with this? It's a transcription factor. I know what they are, but I don't know how to prosecute this into something that's going to be useful for for drug therapy. And that was really a huge problem for us for quite a while. We, of course, also wanted to know, well, this is a mouse. What about humans? I don't really want to cure mice. That's not as interesting as curing humans. So we collaborated with a couple of groups at University of Washington at Oregon Health Sciences, and they had these very rare families of people in which half the male children would die within months of birth. And it was clearly genetic. You could map it. Here's one of the trees right here. And they died from what they called IPEX, which was immune dysregulation. Again, diabetes, skin conditions, chronic IBD, et cetera. They looked a lot like the scurfy mouse. And so it was very easy. Get DNA from patients and family members, sequence the FOXP3 gene. The gene became called FOXP3. I'll get to that in a second. Sequence the gene and find mutations, and we did. So every one of these we looked at, we found a mutation in the coding sequence of the gene. And so we could now identify these families. There were at the time were 200 worldwide. I'm not sure what the number is today, but it's a very small number of individuals who have this. So this is what the protein looks like. This is, I'm sure, somewhat out of date, but it's close enough. So there are repressor domains. This is predominantly a repressor as a transcription factor, although it does depend on what it co-binds to, and so there are activation examples as well. It has a fork-head binding domain to hit the DNA, and a zinc finger and a leucine zipper for dimerization with other transcription factors. And there are many other transcription factors, as you can see, it binds to. Again, this isn't really even a complete list of the full transcription factors. Unlike Drosophila genetics, you do not get to name your own gene, which was very disappointing to me at the time. So this became the third member of the FOXP family, so it's FOXP3, which was depressing. You do get to name your protein, so we called it scurfin. Not very creative, but at least we got to name our protein. So that we felt good about that. We did make the transgenic animal, so I've told you, like, this is the gene, but I haven't actually proven that to you. So we made an animal that expressed the wild type gene, crossed it back into the mutant line, and when you cross them together, if an animal is carrying the mutant gene alone, you see a lymph node that is that dot in the middle, that really big white dot. But if it's mutant but also carries the transgene, you have a normal sized lymph node. And so Mary and I were the two, when we got the transgenics, we said, okay, I need to do the dissection. I need to see this just to make sure this is correct. So we actually sat down at the bench and did the whole dissection, which was terrifying to our technicians. But it was fine. So we got the right gene. That was very good, and we were very excited about that. We did a couple of other things that are interesting. I don't know if they're useful, but they're interesting. But they were helpful to us in understanding tolerance a little bit. So we also made some mice that expressed the transgene only in the thymus. So, T cells all develop through the thymus. And there was the possibility that this lineage, this functional lineage, was generated during thymic development, and then it went off and did its thing, which is what most T cells do. And so we made ones that only expressed it in the thymus. But if we crossed those to the mutant animals all got sick. So you needed to have the FOXP3 gene on in peripheral T cells in order for this to work. And so that told us that this was involved in peripheral tolerance. It wasn't just the filtering process that the thymus does to get rid of self-reactive cells. This was an active process that occurred in the periphery, which again is more interesting to us in terms of thinking about therapeutic development, but it also really highlighted the role of peripheral tolerance. But it still didn't tell us how any of this worked. So a scientist in the group, Rolly Kotri, was trying to figure out where is this gene expressed, what are the cell populations, so we began to isolate as many different populations as possible. One of the reasons I had prioritized this as the bottom of the list is that if you looked in tissues, just by a northern blot analysis, this transcript was expressed in all tissues at very low levels. And that just didn't make any sense to me. It's an immunological problem, not a tissue problem. That's silly. It can't be that. Well, it turns out when you looked at this subpopulation of cells, which was described by Shimon Sakaguchi in 1995, and he labeled regulatory T cells, which is largely a rebranding of suppressor T cells, but with a little more rigor. When you looked at those populations of cells, that's where all the transcript for FOXP3 was. It was only found in these regulatory T cells. So Shimon had shown that those cells can prevent autoimmune diseases in a bunch of a bunch of settings in mice using transfers and by other means. And so they could control autoimmune disease. And now we know that this transcriptional protein is the regulator and is absolutely required for the generation of those cells. And I'm not going to show the data, but the scurfy mice have none of those cells. It's actually a little bit hard to demonstrate that data. It gets tricky. But they don't have those cells at all. Also true in the human. You only find this protein in those cells in the human. So now we knew that, that this was the controlling gene for this regulatory T cell, and those cells were absolutely required to maintain peripheral tolerance in all normal individuals. What we also learned, though, from the transgenic animals is that if you overexpress this protein, those cells become hypo-responsive. So you can see on the top, this is just a response to a titration of stimulation. The mutant animals respond at a lower concentration, and they respond to a much greater intensity. The wild type is in the middle, or normal litter-mate control is in the middle. The transgenic animals, and we have a bunch of lines that had varying levels of transgenes, they always were less than even the wild type. So this really repressed the response of any normal T cell as well. So we essentially could convert normal T cells into regulatory T cells, and that's actually shown here. This is an assay that people use to show the activity of regulatory T cells. And what you see here is on the left side, so the blue column is simply, I love looking at old, like I don't even know what this program was. It's hilarious. Anyway, but the blue column, or blue cylinder, I guess, is the response of a normal cell. If you add regulatory T cells, which is the far bar on the left, you can see that response goes down. If you add other types of T cells, the response actually goes up, which is what we would predict. That's all fine. But that one on the left is the measure of regulatory T cells. When you use transgenic cells, any T cell can inhibit this response. So now we can impart this regulatory function on virtually any T cell that expresses the transgene. And so this really does look like a master regulator for regulatory T cell biology. So that's what's on here, and I think I've said everything that's on this slide. And that was really important to us, and we also knew it was important more globally. So we postulated back in the, 2002, I think this is from, that if you could, on the left side of this, create more of these cells, have these cells become more active, have them become, more suppressive or home better, et cetera, you might be able to treat disease. Now, all those things in the green boxes were postulated things we could do. One of them actually seems to work, but most of them don't actually seem to help us very much. On the flip side, if you could inhibit these cells in the context of a tumor, you might get a better tumor response. And those are clinical studies ongoing today using very different technologies. But I think, we knew this was true back in the early 2000s. We knew this was really important. We just didn't know how to do these things. And it turns out most of them were things we really weren't able to do with the tools available to us. And so then what happens is you unleash this on the immunology community, and about a decade of work happens in which they figure out, well, these cells have a different T cell repertoire than other cells. They require different signals to survive and to expand. They use multiple different mechanisms to inhibit lymphocyte activity. They're involved in tissue homeostasis and tissue repair. They make factors that actually help repair your tissues following an inflammation event. In fact, that's why they're in the tissues. If you look at these cells from liver versus these cells from muscle versus these cells from adipose tissue, they're all slightly different. They use different metabolic programs. They have different T cell repertoires that they see. They have different gene expression profiles, and they're tuned to the tissue that they're in. So they're very important for just maintaining normal tissue homeostasis, et cetera. The other thing that happened during these years is people learned to isolate them in the human. So you notice I didn't really show much human data because it's very hard to find these cells in human. But we learned how to do that in the human. And so people began to think about, well, can I just grow up a bunch of these cells and give them back to somebody who has immunological diseases? And those were done. Studies were done. People found biological responses. People found that they were safe. The cells could stick around for a surprisingly long period of time, but they didn't have any real clinical benefit, nothing significant in the way they actually use them. And so there's lots of potential reasons for that. So if you flash forward to 2019, late 2019, right before the pandemic, so it was a great time. I and 3 of my colleagues started a company, Sonoma Bio, to actually try to use modern technologies here. So now we knew how to isolate them and grow them, but we could also engineer them. So we could say, look, let's make them more active in a particular setting under particular conditions. And let's just amp them up so that they're more focused on a particular disease. And so we did this. There's about 3 or 4 other companies that are doing the same thing. I'm going to talk about this data because I have it, and it's exemplary, but it's really not fundamentally different from what a bunch of other groups are doing in most respects. And you can see all the reasons why we actually think the cell biology and the cell therapy makes more sense in some respects than doing the targeted therapies, which is what we've, as a community, have done for many, many years. There is Enbrel and Humira and the Stelara. There's antibody things that take out a slice of the immune system and work fairly well for a very small segment of some population of people with disease. But it's not broad. What we like is the idea of using what evolution gave us to reset this system. So the way I think of it is, I don't have to be smart about the biology. That cell's got hundreds of thousands of years of being smart. I can just use it. And anything I can do that's useful where I don't have to be smart is really, really good from my perspective, because I'm wrong a lot more than I'm smart. So we set this company to do this, and this is built on some of the polyclonal data. The process is pretty simple, although it is it has its challenges. The little, you probably can't see this, but the little red box up there, do I have a little, does my thing work? Can I do this? There we go. So in humans, these are the cells we care about. And you can see that it's a small population and it's not incredibly distinct. And so you have to figure out exactly where to put this gate. So we run these through a cell sorter and we isolate just these cells away from these cells. And if you don't do it right, these cells will outgrow these cells really easily. But if you do it right, you can end up with a whole lot of these cells at the end of the day. And this is this process here. We get a whole bunch, put them back into somebody. And what we can now do is we can engineer these. But originally, people just isolated these, and again, they were safe. They could stick around. I'm going to show this just for a second, but they could stick around, and they were stable. So the epigenetics of these cells are really important. They're epigenetically distinct from other lymphocytes, and that provides, I think, a large measure of stability. So a year after these cells were infused, you can see they're infused. These are different doses. It doesn't really matter. There's a lot right after you infuse. They begin to disappear over time. This is a year. But at a year, these patients still had easily detectable cells here. And we measure these cells by deuterium. So we labeled the cells with deuterium before we put them in. And if you isolate different populations of lymphocytes from the blood, the only place you find deuterium is in the cells that we grew up. So that was very nice. And you can find them a year later. And these cells never become any of these other populations. So they seem very stable, and they can live for quite a while in the blood. So we decided to focus on rheumatoid arthritis. Again, I'm not going to belabor this, but there's some data in arthritis in which suggests that these cells are somewhat defective. They're not gone. They're not horribly messed up, but they're slightly defective, and the level of their defectiveness tends to correlate with the level of disease within the arthritis patients. So we thought this was a good place to start. We could do biopsies, we believed. You'll see a little bit of data at the end. It was much harder than we thought. But we could do biopsies to look at the cells and see if they got to the tissues and did what they're supposed to do. So we wanted the translational side of this to work as well. So we could expand them on the right. They still had high levels of FOXP3. They still looked like good regulatory T cells. That was all very nice. We decided to engineer them to recognize a protein that's found predominantly in the rheumatoid joint. In this case, it's citrulline proteins. So this is very common and is somewhat diagnostic, actually, for a number of rheumatic diseases, but most prevalently rheumatoid arthritis. It's found in a number of other places, but it's not typically found in most normal tissues. And it's a process by which arginine gets converted to citrulline based on enzymes that are largely produced by neutrophils, but other cells can produce these factors as well. And so you could see there was a lot of citrulline proteins there. We created a CAR. So a chimeric antigen receptor, very similar to what people do in oncology. The signaling domains are different because we're not signaling into a CD8 cell, we're signaling into a regulatory T cell. They require different signals to survive and proliferate and become activated. So the signaling domains are different, but the principles is virtually the same. We put a tag on these so we could find them as well. And then the idea is to go into rheumatoid arthritis patients with these cells and ask, can we treat the disease? The cells respond to various citrullinated proteins. The one that's most commonly used is vimentin, but in fact, there's cross-reactivity of this particular receptor, which we got from a collaboration with the Karolinska Institute. So it sees fibrinogen, keratin, filigrin, a number of other proteins. Some of these are purely intracellular proteins, the histones, for example, so that's not going to be useful for our CAR. But it turns out vimentin, filigrin, some of these other proteins actually get deposited in the extracellular matrix and on the outside of cells, which is key for our CAR T cell to recognize those proteins and become activated. And in fact, you can even see activation of our CAR T cells if you look at synovial fluid. And so that's on the right here. So I'm not sure how we got fluid from non-RA patients, but from RA patients, you can see that the fluid there would turn on our CAR T cell. Now, there's not a lot and it's not great, but it's certainly well above background, and it's very consistent as well. And so this is a somewhat soluble version of these citrullinated proteins. If you spin that serum really hard and clarify it, a lot of that activity goes away. So I think it's really multimeric in the synovial fluid, which again would fit with what we know about this. When we engineer them, the product is called 77101. The company is Sonoma Bio because Jeff Bluestone has a house in Sonoma. And so the 77101 designation, all biotech companies have names for their stuff. That's a grape clone. That's a Pinot Noir clone. So that's how we do it. Got to have a little bit of fun while you do this. So, so when we generate our product, they look like very good regulatory T cells. Actually, in some measure, they look like better regulatory T cells than the cells directly out of the blood, which is the top three rows on this. The bottom three are product, different donors, and a number of different markers there to assess them, their Treg-like status. So they look very good. That's great. They also seem to be very stable if you stick them in pro-inflammatory cytokines. So these cells are theoretically going to go into a site that's inflamed. So lots of IL-1, lots of TNF, other cytokines. So we want to make sure that the cells are stable and active. In fact, they are. The two columns on the right there are effector T cells on the far side of that graph. And you can see just by color, they're very different from the four columns on the left. The four columns on the left are regulatory T cells, either unexpanded or expanded. And you can see they're very different. And that's true no matter how you want to look at this. And I think I'll show you another plot on that in a second. Here, actually. So this is, again, an analysis of effector cells. In the first column, regulatory cells. In the second column, these are just freshly isolated cells out of the blood. If you expand these cells, they look similar. They maintain this same fingerprint identity. And so that's, again, super important for us as we think about developing this as a drug. The other thing that I'll point out is that this is busy, and I'm not going to go through this line by line. Don't worry about it. Again, effector cells in the group on the left, and you can see regulatory genes are on the bottom half of this. They're mostly purple. Effector genes are on the top half. They're mostly red. That's good. We isolated the right cells. Regulatory cells in the middle, it flips. Blue stuff at the top, mostly red stuff at the bottom. But if you look at effector cells that you've transduced with FOXP3, so can you convert an effector cell into a regulatory cell just by giving it FOXP3? The answer is sort of. So not really. You can see that it's somewhere in between, and there's a bunch of cluster analysis that does the same thing. So now, is that good enough to make it a suppressive cell in a clinical setting? I don't know. There's a company that's trying that, and we'll find out. So I don't know what the answer to that is. They might be good enough, but they're not the same. And I think that's important. We use what I like to refer to as native or natural regulatory T cells. We developed a process to manufacture these, which seems trivial. One of the big steps was thawing the cells. So it seems like, lymphocytes, we thaw T cells all the time. Treg cells don't thaw nearly as easily as a standard CD4 cell. I have no idea why, but they don't. So we figured that out, and we figured out how to put the lentivirus in them and transduce them and what day and all of those sorts of things, when to stim. Hello. Okay, let me go back. Oh, come on, go back. Seriously. Why is it we talk to inanimate objects? Okay, so we developed a clinical study. It took us a while to convince the FDA this wasn't silly. And so 3 dose levels, very standard clinical trial, which is screen the patients. During the pre-treatment period, we would get the cells, expand them, and it's probably a 6-week protocol before we're ready to dose. It's something like that. I don't know the exact number. And in most cases, get a pre-treatment biopsy. Now, I went into this assuming, oh, rheumatologists biopsy people all the time. They do not. Not in America. So we were using ultrasound-guided biopsies. We actually sent people from Stanford and University of Colorado and Tufts to Oxford, England, to learn how to do an ultrasound-guided biopsy, because they don't use them. They just throw drugs at patients. They don't actually look at the joints anymore. I was stunned, actually. But okay. So we try to get biopsies, and we're getting better at it now. But 3 doses, and look, obviously it's a safety study, and that's really what we're trying to do. But of course, we're trying to get as much information out of these patients as possible. And I'm going to show you 3 patients. So first dose level, second dose level. So patients 1, 2, 3, 4, 5, 6. You can see the characteristics of the patients. They're all females, mature. So there's no kids involved in this anywhere. 42, I think, is the youngest person. The thing I've highlighted in the bottom is that we had a criteria in which they had to have been on and progressed on at least 3 DMARDs or 3 mechanism of action drugs. So it had to be a TNF antagonist, rituximab, and a JAK, or pick your 3. One of them's always going to be an anti-TNF, but one of the others. So they've all been on at least 4. One of these patients has been on 6 different mechanistic drugs, and they've been on a total of 12 different drugs. So these patients have very progressive disease and have been very unresponsive in general to this. The other thing I will point out here is the level of inflammation is really quite variable. So if you looked at the swollen and tender joint counts as one measure of this, you can see the first one, it's only 5 out of 28, 7 out of 28. Now, I'm not suggesting that that's not bad, but it's not as bad as 22 out of 28, which is really bad. And so the amount of inflammation that's detectable at the time of enrollment in this is really quite variable, which was something we weren't really expecting, but it's what we ended up with. So here's the response. So again, I'm going to show you one. There's a lot of other data, but this will suffice for right now, which is the yellow orangey things on the left is the lower dose, the blue and black thing on the right is the higher dose. You can see following the dose, most patients have a reduction in inflammation. And so it's swollen joints on the left, tender joints on the right. And whether you measure 28 joints or 66 or 68 joints, the conclusions are roughly the same, which is in most patients you'll see a response. Some it levels out, some they come back up. Slightly better response, I would say, in the higher dose level, but it's early days. We haven't gotten to the third dose level yet. And again, this is still fairly early numbers of weeks. This is only out to 24 weeks. And obviously there's more data that I don't have at the moment to see how long the durability lasts, et cetera. No safety issues in any of these, so I'll just point that out. I don't think I even put a slide in there, but there's no safety issues whatsoever in any of the patients. So that's obviously encouraging. We weren't expecting any, but so it's why you do the study, and it's why you look for these things. If you looked by ultrasounds, so this is pre-treatment on the top. You can see orangey red showing of inflammation decreased substantially 4 weeks after infusion in this particular patient. We don't have this on all the patients, unfortunately, but we're getting there as we enroll more and more patients. If you do the histology, you see a very similar thing. The Kren score, I'll point out, 9 is the highest value you can get in the Kren score. So participant 5 went from 9 to 1, which is awesome. But the participant 4 went from 3 to 2. Less awesome. Right trend, still looks good, but they weren't as inflamed. and so you didn't have it nearly as much of an effect. But at least it's going in the right direction. Inflammation is going down, et cetera, on this. I will parenthetically state that there are some measures that we use which are less quantitative. Can you open a jar of pickles? Turns out there are people who could not open a jar of pickles for 5 years, but now can open a jar of pickles. If you like pickles, that's an important thing. So that was good. One of the participants came back in at a study and said, God, I'm aching today. I feel really sore. And the PI said, oh, I thought you were doing well. What happened? He said, well, I went for this run yesterday. I'm like, when was the last time you ran? Oh, 5 years ago. And I get this, and I'm like, don't let them run. What are you doing? No, they can't run. Stop it. I mean, of course, it's good that they could run. It's just confounding for me. But that's my problem. If you look for, in some of these patients, we do have the on-treatment biopsies, and we use in situ hybridization to look for the presence of our CAR. And I know you can't see this, but yes, it's there. Not a lot of cells, right? But there's not a lot of inflammatory cells in general. The CAR is always associated with a FOXP3 CD4 positive cell. So again, that's really good. Not all the cells that are CD4 in this joint are our cells. So there are still some endogenous native responding cells. There are some CD8 cells in some of these as well, and those obviously are not our product. So there are still other T cells in some of these settings, but our cells are certainly there. So we're pretty encouraged by this. Again, very early days. Other companies are doing trials as well. I don't think there's been a safety signal in any of those trials that I'm aware of in talking to people and seeing what people have published. So I think the principle is that these cells will be safe. And so now we're at this future point where we understand a lot more about what controls peripheral tolerance, why most of us don't have autoimmune diseases, and some of us do in some cases. And I don't think regulatory T-cell biology is the answer to all autoimmune diseases. I would never state that. It will not be the cure to all autoimmune diseases. But I am very intrigued with the idea of using a cell that has multiple intrinsic capabilities of controlling inflammation and can provide tissue repair and tissue homeostatic mechanisms at the same time. Most drugs don't do that, right? They stop something and hopefully everything else takes over. And in some cases it works. But here we're providing back many things. The phrase some people use is a polypharmacy. Now it makes it more complicated, but I also think it makes it more natural at some level, and I think it's more comprehensive. So yes, it may be complicated, but I think it's also comprehensive in many respects. Okay, so I think I'm done. Yeah, I'm done. All right, so let me tell you a little bit who did this. So David Gallus was a mentor and an amazing scientist. Came out of USC, I'm sorry. But fantastic person involved in the Human Genome Project and became the first CEO of Darwin Molecular, and he was our connection to Oak Ridge National Labs, which was great. Mary on the upper right, who I shared the prize with, is a longtime friend and truly stellar person. Roly Khatri on the bottom right is one of the more undersung people in this. She's the first person to know that FOXP3 controlled regulatory T cells because she came in and told me in my office. She'll never get the amount of credit she deserves. Steve Ziegler is really the first person who saw the scurfy mouse and recognized that it would be really important. He and I worked together at Darwin. He's now at Benaroya Research Institute. You see the geneticists and other folks up there as well. The folks at Sonoma, Jeff Bluestone is a longtime friend and CEO and force of nature in general. Joe Bialke is one of the first hires we had, or actually I think he technically was the first person at Sonoma. Somehow he started before Jeff, and I don't understand how that worked. But anyway, he's now the CSO at Sonoma, and the programs are in extremely capable hands. One of the best experimental scientists I've ever known. I should mention Lars and Vivian at the Karolinska Institute, who were instrumental in helping us figure out what antibody to use to create that CAR, and they have incredibly deep understanding of the role of citrullination and citrulliнаted proteins in these diseases, and they've been fantastic collaborators as well. And my final slide is always the most important. People, the mice. You know, I spent a lot of time studying mice, and people ask, are mice good models and stuff? And I spent a lot of time consulting for various groups and stuff. And I said, look, the mouse has been proven to be a phenomenally accurate representation of how the immune system works in humans. It's not perfect, right? There definitely are differences. But all the basic principles of immunity in the mouse are pretty much mirrored in the human. The mouse is a terrible model of human disease. It's acute. Size matters in some cases. It's induced. We put tumors in the subcutaneous areas, which is not where the tumors live. It's just a terrible model of human disease. But it's a really good model if you want to study the principles of immunity. And it's not unuseful for human disease. I think it is, but I think we overemphasize it far too much as a model for that. But they're also really cute. So, all right, happy to take questions, comments. Thank you very much for listening.